In this program, we will try and retrace the origins of man. We will then look at various dating methods and conclude with palynology, the science of fossilized pollens. For a long time, paleontologists dated the great split between man and apes at 12 million years ago. But recently, geneticists and biologists have suggested a far more recent date. The split would only have occurred between 3.5 and 5 million years ago. Now, this gave rise to a general outcry from the paleontologists. But in spite of the differences of opinion, a consensus seems to have been reached on certain stages in man's evolution. Let's start with the Australopithecines. Archaeologists and paleontologists are retracing the origins of man by studying carved stones, bits of bones, and rock paintings. Today, most of these researchers agree on a genealogy that sets the origins of mankind back to the Australopithecines, which were man-like primates that inhabited southern and eastern Africa some 4 million years ago. They were skilled at climbing trees, and their fossils suggest that they were evolving into bipedal walking. Then evolutionary change, such as the increase of brain size, led to the emergence of the Homo genus 2.5 million years ago. Homo habilis, or handyman, was its first representative. The first tools are ascribed to him. His descendant, Homo erectus, was the man of discoveries and travels. About 1.5 million years ago, he left Africa to migrate to Europe and Asia. The earliest use of fire is attributed to him. Then in Europe, some 100,000 years ago, Homo erectus evolved into a sort of primitive Homo sapiens, the Neanderthal man. A great hunter, the Neanderthal man also buried his dead. In Africa and Asia, the evolution of Homo erectus continued, leading to modern man, Homo sapiens sapiens, with the Cro-Magnon man as its prototype. 35,000 years ago, the Neanderthals disappeared, leaving the field clear for Cro-Magnon and his descendants, us. Before this hypothesis on the origin of man could be made, countless archaeological excavations were conducted, mainly in Africa, Asia, and Europe. Such places as rock shelters and caves are conducive to the preservation of the traces of man's passing. One such place in Europe is Tottevel, where the bones of the oldest European male were uncovered. To get an idea of how prehistoric man lived, archaeologists delimit an excavation area of about 1 square meter. From within that space, they expose the rocks, bones, tools, and other objects, without, however, removing them from their locations. To do this meticulous work, they use awls, brushes, and trowels. These areas are then recorded by photography, drawings, and cast reproductions. The pinpointing process then begins. That is, the exact positioning of the objects within the site, as well as their positions relative to each other. This invaluable information on the relationship of the various objects tells us a great deal about the lifestyle of prehistoric man. For example, the juxtaposition of animal skeletons and tools might well be an encampment where primitive men cut up game killed during the hunt. Finally, all the uncovered objects are washed and numbered. In the laboratory, researchers compare the bones discovered on the site with the anatomy of present-day man. Their comparisons allow us to conclude that the Tauntable man, a Homo erectus, had a receding forehead and a more protruding face, compared with modern man and the Cro-Magnon man, whose face is set under his cranium. The Tauntable man has other characteristic traits, a thick roll of skin above the eyes, as well as a chinless jaw. With these features, he looks more like an ape than present-day man. Also, his long tibia and femur bones are more massive than modern man's, which makes him a far more robust and sturdy walker. By studying with an electronic microscope an individual's teeth and their wear, the experts can tell what he ate. The teeth of carnivores, for instance, present regular vertical grooves, whereas herbivores have more complex grooves in all directions. Further, paleontologists examined the pathologies affecting bones found in excavation sites. The pathologies have shown, for example, that in certain species of prehistoric man, there existed a social order in which their disabled were cared for. However, in spite of all the findings and research to date, many questions about the origins of man remain unanswered. Archaeologists' work consists largely in establishing a chronology. In the old days, the dating methods they relied upon were only relative. They were based on the fact that when terrain has not been subject to upheavals, the layer closest to the surface is the most recent. It was therefore possible to roughly assess how old the various layers were, as well as the fossils they contain. These methods are inaccurate. In some areas of the world, there are no sediments. Elsewhere, the ground has shifted. Fortunately, today we have absolute dating methods, the commonest of which is carbon-14. In archaeology, the dating of objects, bones or tools, is of utmost importance, since it is the basis on which the reconstruction of the course of history is made. An idea of one of the fathers of the atomic bomb, Willard Libby, has allowed science to come up with one of the most useful instruments to study the past, carbon dating with carbon-14. Libby demonstrated that radioactive substances are formed by the constant bombing of the Earth by cosmic rays. He understood that when these rays go through the atmosphere, they strike atoms of nitrogen. The nitrogen atoms lose a proton while gaining a neutron, and by the same token, become radioactive carbon-14. Carbon-14 quickly combines with the oxygen in the air to produce carbon dioxide. In the form of carbon dioxide, radioactive carbon-14 spreads out broadly into nature. Plants are the first to absorb it, converting it through photosynthesis and incorporating it into their tissues. This is how all the living things that consume plants ingest carbon-14. Carbon-14 atoms are unstable. They decay. That is, they spontaneously convert back into nitrogen. However, as long as an organism is alive, the carbon-14 atoms in its tissues are continuously renewed through breathing and eating. When the organism dies, the carbon-14 atoms that decay within it are no longer replaced. Their number decreases by half every 5,570 years. So by measuring the amount of carbon-14 that remains in a bone, for example, and comparing it with the amount contained in a bone belonging to a similar living organism, the specialists are able to calculate how long it has been since the organism died. All organic matter with carbon in its composition can be dated by the carbon dating technique. It is common practice to date wood, coal, cereals, materials, bones, and seashells using this method. The quantity of carbon-14 in a sample of organic origin can be measured two ways. The first consists of burning the samples in the presence of oxygen to convert them into carbon dioxide. The gas is then put into a radioactivity detector that counts the level of radioactivity due to carbon-14. This technique allows the experts to date objects that are up to 40,000 years old. The second method, called accelerator mass spectrometry, enables them to count the atoms of carbon-14 directly instead of measuring the level of radioactivity. The main advantage of this new technique is that it can date samples having very little carbon. Indeed, it requires 1,000 times less carbon than conventional radioactivity meters. When archaeologists want to determine the age of objects having very little carbon in them, such as pottery or prehistoric flint tools, they use thermoluminescence. Rather than considering the disappearance of a radioactive element, like carbon-14, from an object to be dated, they measure the effect radioactivity exerted on the object over the course of time. For instance, a flint tool is subjected to two types of radiation: the radiation emitted by the radioactive elements in the flint itself, and the radiation from the ground in which it was buried, since it was used by prehistoric man. These radiations pull electrons away from the atoms of the mineral that flint is composed of. The electrons are trapped by structural flaws in the mineral. They therefore gradually accumulate in these traps over the years. When the tool is heated at a temperature of 500 degrees Celsius, the captive electrons are suddenly released. They then emit a luminous radiance called thermoluminescence. When a tool made of flint was heated by prehistoric man, around a fire, for example, the electron reserve escaped, going back down to zero. So the electrons would start piling up all over again. The longer this process goes on, the more electrons pile up, and the more intense the thermoluminescence measured in the laboratory. This is how the experts determined the period when certain flint tools were made by man. Thanks to thermoluminescence, we can date objects going back 200 to 300,000 years. It goes way beyond the carbon dating technique. Any progress we make in our knowledge of the origins of man depends upon the development and improvement of our dating techniques. Researchers' work does not merely involve dating fragments or flint stones. Their ambition is to come up with an overall model of the conditions of life that existed in the past. To do this, among other things, they study the climactic conditions that prevailed through the ages on our planet. These studies are part of a specific scientific field called palynology. Who would have ever imagined that certain flower parts could be the undying witnesses of a distant past going back several hundred or several thousand years? Well, it's true. Under certain conditions, the pollen grains produced by all flowering plants can be preserved almost indefinitely. A science called palynology is very much interested in remains like these that have accumulated in lakes, peat bogs, and estuaries. Pollen grains are the reproductive cells of flowering plants. They play the role of male sexual cells. Carried by the wind, insects, or by some other means, these grains have to join with the ova of a flower of the same species in order to fertilize it. Spores do the same for fungi, moss, and ferns. These reproductive cells, pollen and spores, are only a few microns wide. They are enclosed in an envelope consisting of 3 very resistant chemical substances. Chitin, also found in the composition of the exoskeletons of certain insects. Cutin, which coats bay leaves, for instance. And finally, sporopollenin, composed of a substance very similar to lignin, the basic component of wood. Since this envelope is highly resistant to chemical and meteoric agents, it ensures the pollen grains virtually perfect preservation when they settle in spots away from the oxygen in the air. Moreover, every plant species is characterized by specifically shaped pollen grains. The thousands of spores and pollen grains the plants produce every year are scattered in the surrounding areas by the wind. Once they have come to rest, they integrate into the sediments year after year. By making an inventory of them from sediments of a known geological age, the scientists are able to come up with a fairly accurate idea of what the surrounding vegetation was like over the ages. Further, since vegetation constantly reflects climate, what their analysis of the pollen tells them about the vegetation of a given area also informs them of the region's climatic conditions. To find out about the history of the vegetation and climate of a region, the palynologist begins by drilling. He extracts from the soil cores of sediment about 50 centimeters long. In the laboratory, he extracts the pollen from the cores. By chemical dissolution and degradation of density, he removes the mineral and organic matter fastened to the spores and pollen grains. He can then examine and identify the isolated spores and pollen grains by microscope. A statistical analysis of the various pollen grains and spores enables the researcher to estimate the relative proportions of each plant species. The image he obtains from his analysis will help to determine the climate that existed when this particular vegetation flourished. Our knowledge of past climates is based chiefly on comparisons drawn today between pollen, vegetation, and weather conditions, such as temperature and precipitation. So it's in reference to what they observe today that palynologists manage to determine the history of a region's vegetation and climate. The palynological study of a region can help to discover many things. For example, following the thaw of the glaciers, the vegetation was primarily made up of buttercups, herbaceous aquatic, and floating plants. Then the first trees appeared, among them the juniper and the birch. As the soil became richer with organic matter, these pioneer species were replaced by forest trees like oak and hazel trees. These trees thrived in the average temperatures that lasted for 4,000 to 5,000 years. Then a cooling caused vegetation to evolve toward the beech grove and the fir forest. Finally, when man emerged, the forest was largely destroyed to make way for grain crops. Palynology manages to reconstitute these kinds of evolutionary cycles. Not only does it allow us to learn more about the physical surroundings in which prehistoric man evolved, it also gives us an indication of things to come, like future climate conditions. In all excavation sites, large quantities of bifaces, that is, two-edged flints, are found carved in an almond shape. Also, small stones covered in tiny, irregular grooves, and others decorated with colored lines. One can only wonder about such a preoccupation with symmetry and beauty, when a plain old fragment of stone would have done just as well.